For decades, testing catch-all treatments against a heterogeneous disease resulted in failed trials, where biological decay was viewed as an irreversible absolute. WashU's chart shows 25% of patients achieving complete disease stabilization, with functional scales revealing improvements in grip strength and respiratory capacity. Conversely, UC Davis engineering bypasses biology, translating neurosignals into speech at 62 words per minute with 97% accuracy. These specific results from WashU and UC Davis demonstrate that the progression of ALS can be physically halted at the molecular level and is technologically bypassed when biology fails. This inflection point in patient outcomes is not the result of a single molecule or a lone scientific discovery. It relies on the convergence of three distinct, highly specific technological and medical pillars. The first pillar is upstream diagnostics, utilizing multi-omics to stratify patients before symptoms emerge. This feeds the second pillar, downstream molecular intervention, where therapeutics target specific RNA and TDP-43 dysfunctions. The third pillar addresses late-stage decline, using high-bandwidth software to restore environmental control. Artificial intelligence acts as the middleware linking these three pillars. It ingests the diagnostic data, matches the precise molecular drug, and calibrates the hardware, turning isolated breakthroughs into a cohesive, predictive care protocol. Establishing this protocol requires moving beyond isolated genetic mutations and tracing the disease's origins directly into the gut-brain axis. Researchers at Case Western identified that specific gut bacteria produce harmful glycogen sugars. These microbial metabolites bypass normal biological barriers and spark aggressive immune attacks on motor neurons, a mechanism observed in both ALS and frontotemporal dementia. Pinpointing this gut trigger provides an entirely new vector for treatment. Modulating the microbiome presents a highly accessible, early-stage intervention point before neuronal damage becomes severe. Blood analysis provides another critical layer of intelligence. Mass General Brigham identified a highly accurate plasma-based protein signature that distinctly flags the presence of ALS. This specific protein panel detects molecular changes up to 10 years before physical symptoms manifest. Biogen is already utilizing this pre-symptomatic detection window to enroll early SOD1 carriers in global clinical trials. With early detection secured, patients can deploy targeted nutraceutical adjuvants. Combining trace minerals and highly bioavailable curcumin directly addresses known biological failure points, like SOD1 metallation defects. Tracking gut metabolites and plasma proteins a full decade early expands the therapeutic timeline. Clinicians can intercept the pathology before irreversible motor neuron death occurs. Even with early detection, genetic profiling has historically hit a severe bottleneck. Over 90% of missense mutations leave patients in genetic limbo, classified formally as variants of uncertain significance. Computational models are clearing this gridlock. By combining predictive algorithms with hard experimental data, new pipelines systematically reclassify these ambiguous genetic markers. This hybrid approach successfully resolves 75% of previously unknown variants, achieving an error rate of less than 1%. Once a variant's pathogenicity is confirmed, the genomic data feeds directly into trial matching algorithms. This system identifies specific high-priority patient phenotypes, such as individuals with rapidly progressing disease profiles. This stratification mechanism actively populates trials like Prevail's. By filtering for early, fast-progressing patients, researchers drastically improve the signal-to-noise ratio in their clinical data. Resolving the genetic bottleneck upgrades raw DNA sequencing. It shifts genomics from a passive diagnostic readout into an active routing system that places patients into highly targeted Phase 3 interventions. When patients reach those trials, the primary biological target is clear. In 97% of all ALS cases, the central pathology traces back to the dysfunction of a single protein, TDP-43. The failure is structural. A healthy paired TDP-43 dimer normally regulates RNA. Under stress, it breaks apart into chaotic monomers that clump into toxic aggregates. The loss of healthy TDP-43 triggers a cascade of errors called cryptic polyadenylation. Without the protein acting as a regulatory supervisor, rogue segments of code slip into the RNA sequence during splicing.
We observe this exact failure in the PKN1 gene. The insertion of a cryptic exon here directly results in the production of the neurotoxic N207 peptide. Understanding this mechanism clarifies the therapeutic objective. Instead of merely attempting to mop up dead protein aggregates, researchers are deploying molecules designed to directly correct the underlying RNA splicing errors. For the aggregates that have already formed, pharmacology offers a mechanical solution, using drugs engineered to act as molecular janitors. Researchers at the Mayo Clinic identified the KPNB1 chaperone protein, providing a direct, druggable handle to dissolve pathological TDP43 formations. Small molecule drugs enhance this naturally occurring chaperone. By actively clearing the intracellular debris, these enhancers restore actual neuronal function, rather than simply slowing the rate of decline. Validating these clearance mechanisms clears the way for advanced gene therapies. Antisense oligonucleotides use specialized viral vectors to deliver precise genetic instructions directly into the central nervous system. The 3-year tofersen data proves this logic holds up in practice. By successfully targeting and degrading mutant mRNA, that trial demonstrated that precision molecular engineering works in living human nervous systems over multi-year timelines. Once artificial intelligence identifies the precise genetic failure point, clinicians possess the specialized chemical tools required to enter the cell and manually correct the biological error. When a patient's biological systems degrade beyond the reach of molecular intervention, the clinical strategy pivots entirely to neural engineering. Brain-computer interfaces are transitioning out of bespoke experimental lab setups and into standardized, mass-produced consumer hardware. Automated robotic surgery scales the deployment of ultra-thin cortical implants, packing tens of thousands of recording electrodes onto the surface of the brain. Parallel engineering efforts bypass open surgery entirely. Endovascular stentrodes acquire high-fidelity neural data by navigating directly through the patient's existing vascular network. Across multiple architectures, the physical acquisition of high-resolution neural signals is a solved engineering problem. The hardware is now commoditized. With the hardware standardized, the true value of a brain-computer interface lies entirely in the software layer running on top of it. Natural language processing models decode the intent. These algorithms analyze raw electrical spikes, translating them into clean text strings and synthesized speech, complete with accurate tone and pacing. Hitting 62 words per minute establishes a rigid new benchmark. That metric is no longer an aspirational ceiling for clinical trials. It is the operational floor for basic patient communication. This software translation layer also bridges the gap to consumer electronics, allowing endovascular implants to natively control augmented reality environments. Deploying this interface code directly rewrites the trajectory of late-stage disease. It replaces the isolation of locked-in syndrome with sustained environmental control and personal agency. Delivering these outcomes at scale requires breaking down the historical barriers, isolating genomics, molecular biology, and neural engineering. This dashboard illustrates a unified modern care protocol. It aligns a multi-omic diagnostic risk score on the left, an exact molecular therapeutic match in the center, and a target functional output metric on the right. Managing this data flow requires continuous computation. A centralized model ingests early diagnostic warnings, optimizes the drug match, and calibrates the digital hardware outputs simultaneously. The rapid commoditization of artificial intelligence compute power accelerates every step of this pipeline, compressing target to trial timelines from years down to a matter of months. Heavy institutional capital recognizes this acceleration. AI-native drug discovery labs and pharmaceutical giants are aggressively securing the massive supercomputing architecture required to process this data. Backed by record federal funding allocations, researchers are deploying highly enriched Phase 3 trial designs. The scientific and financial infrastructure finally matches the biological urgency of the disease. Mapping these biological and digital variables has transformed ALS from an insurmountable biological mystery into a tractable data problem.